Photosynthesis begins with a powerful first step, the light-dependent reactions. This is the phase where plants capture sunlight and convert it into chemical energy, setting the stage for the entire process of food production in green plants. The light reactions, also called the photochemical phase of photosynthesis, involve the absorption of light, splitting of water, release of oxygen, and the formation of energy-rich molecules like ATP and NADPH. These reactions take place with the help of several protein complexes. The pigments involved are organized into two photosystems, Photosystem 1, PS1, and Photosystem 2, PS2. These names are based on the order of discovery, not the order of function. Each photosystem contains light harvesting complexes, LHCs, made up of hundreds of pigment molecules bound to proteins. These pigments absorb light of different wavelengths and transfer the energy to a special chlorophyll molecule known as the reaction center. The reaction center differs in the two photosystems. PS1 has a chlorophyll called P700, which absorbs light best at 700 nanometers, while PS2 has P680, which absorbs best at 680 nanometers. Together, these systems help capture light energy efficiently and drive the initial steps of photosynthesis. In Photosystem 2, PS2, the reaction center chlorophyll absorbs red light at 680 nanometers, which excites its electrons. These high-energy electrons jump to a higher orbit and are captured by an electron acceptor. From there, they pass through an electron transport chain made up of cytochromes. As the electrons move through this chain, they lose energy gradually, a process described as moving downhill on the redox potential scale. This entire process of electron transfer, starting from PS2, moving through the electron transport chain to PS1, absorbs red light at 700 nanometers and finally reducing NADP+ to NADPH+ and H+ is called the Z scheme due to its characteristic shape when carriers are placed on a redox potential scale. Splitting of water. The splitting of water is associated with photosystem 2, breaking water into 2H+, oxygen, and electrons. This process creates oxygen, one of the net products of photosynthesis. The electrons needed to replace those removed from system 1 are provided by photosystem 2 through this water splitting process. The water splitting complex is physically located on the inner side of the thylakoid membrane, releasing protons and oxygen into the lumen. The chemical equation for this process is: this crucial reaction not only provides the electrons necessary for the continuation of photosynthesis, but also releases oxygen as a byproduct, which is essential for aerobic life on Earth. Cyclic and non-cyclic photophosphorylation. Non-cyclic photophosphorylation occurs when both photosystems, PS2 and PS1, work in series connected through an electron transport chain as seen in the Z scheme. This process synthesizes both ATP and NADP, H+ and H+. The electrons flow from water through PS2, the electron transport chain, PS1, and finally to NADP+, reducing it to NADPH+ and H+. Cyclic photophosphorylation occurs when only PS1 is functional. The excited electron is circulated within the photosystem through the electron transport chain back to PS1 rather than passing to NADP+. This cyclic flow results only in ATP synthesis, not NADPH+ and H+ production. It happens in stroma lamellae membranes, which lack PS2 and NADP reductase enzyme, or when only wavelengths beyond 680 nanometers are available. Let's talk about a process in plants called the chemiosmotic hypothesis. Don't worry, it sounds big, but it's actually simple. It explains how plants make energy called ATP during photosynthesis. Let's break it down into 3 easy steps. Step 1. Proton accumulation. Inside the plant cell, there are tiny discs called thylakoids. When light hits the plant, water inside the cell is split. This releases protons, electrons, and oxygen. The protons start to build up inside the thylakoid. Also, as electrons move across the membrane, more protons get pushed inside, so a lot of protons collect in the thylakoid lumen. Step 2. Proton gradient. Now inside the thylakoid, we have lots of protons. Outside in the stroma, there are fewer protons. This creates a proton gradient, a kind of pressure difference. The inside becomes acidic and the protons want to move out. Step 3. ATP synthesis. The protons move out through a special channel in an enzyme called ATP synthase. As they move, they give energy to this enzyme. This energy helps make ATP, the energy currency of the cell. We just learned how plants use light to make energy in the form of ATP through the chemiosmotic hypothesis. Now, let's move on to the next part, where the plant actually uses that energy to make food. This next stage is called the biosynthetic phase or the light-independent phase. What is the biosynthetic phase? This phase doesn't directly use light, but it needs the products of the light reaction. That means ATP and NADPH. These two molecules are full of energy. The plant uses them to take carbon dioxide from the air and turn it into glucose, which is food. So even though light isn't used directly, this phase can't happen without the light. That's why calling it the dark reaction is a bit confusing. It doesn't need darkness, just the products made during light. How did we learn about this? A scientist named Melvin Calvin figured it out. He used a special kind of carbon called carbon-14 to trace how carbon moves in a plant. He found that the first product made when carbon dioxide is taken in is a 3-carbon compound called PGA, short for 3-phosphoglyceric acid. Thanks to him, we now call this cycle the Calvin cycle. Different pathways in plants. Not all plants follow the exact same steps. Some use the C3 pathway, where PGA is the first product. Others use the C4 pathway, where the first product is a 4-carbon compound called oxaloacetic acid. One more thing, this phase can keep going for a short time even after the light goes off, as long as ATP and NADPH are still there. But once those run out, the process stops and starts again when light is back. So remember, it's light independent, but not really a dark-only reaction. Let's look at something interesting scientists discovered: the primary acceptor of carbon dioxide. The early assumption. At first, scientists believed the CO2 would be accepted by a 2-carbon compound. Why? Because the first stable product they saw, PGA, had 3 carbon atoms. So they guessed, maybe a 2-carbon molecule combines with CO2, which has 1 carbon, to make 3 carbons. They spent years looking for this mysterious 2-carbon compound. The big surprise. But surprise, it wasn't a 2-carbon molecule at all. Later research showed that the real CO2 acceptor is a 5-carbon sugar called RUBP, short for ribulose bisphosphate. When CO2 combines with RUBP, it forms a 6-carbon compound, which then quickly splits into 2 molecules of PGA, each with 3 carbons. This explained everything. We hope that you have understood the topic. You can now study chapter notes, watch video lectures, and solve MCQ tests of this chapter on EduRev to ensure you score well in your exams. And that's not all. You also get amazing courses for maths, science, English, Hindi, and much more for NEET. Thank you.